Welcome to another episode of The Cutting Edge, where we delve into groundbreaking scientific discoveries that shape our understanding of health, disease, and the human body. In this episode, we explore the multifaceted roles of the P53 protein, a crucial tumor suppressor, and its implications in cancer and other physiological processes. This lecture is based on a comprehensive review by Colleen A. Brady and Laura Diatardi from Stanford University, published in the Journal of Cell Science. Background on P53. Since its discovery in 1979, P53 has been intensively studied for its role in cancer suppression. Known as the Guardian of the Genome, P53 functions primarily by inducing cell cycle arrest, or apoptosis, in response to various cellular stress signals, thereby preventing the propagation of damaged cells. However, recent research has revealed that P53's role extends far beyond these traditional functions, impacting metabolism, autophagy, and the oxidative status of the cell. P53 and tumor suppression. Analysis of human cancers reveals a fundamental role for P53 in tumor suppression. More than half of all human cancers harbor mutations in the P53 gene, which encodes the P53 protein. The inheritance of a mutant P53 allele predisposes individuals to Lie-Fraumeni syndrome, a condition characterized by a high risk of developing multiple cancers. Studies using P53 in all mice, which develop cancer with complete penetrance, have unequivocally confirmed P53's critical role in preventing tumor formation. Mechanisms of tumor suppression. Stress response. P53 acts as a sensor of cellular stress, responding to DNA damage, hypoxia, oncogene expression, nutrient deprivation, and ribosomal dysfunction. It limits the propagation of cells under these adverse conditions by triggering apoptosis or senescence. Apoptosis and senescence. An ancestral function of P53 and metazones is to trigger programmed cell death, apoptosis, in response to DNA damage, preserving the integrity of the germline. In more complex organisms, P53 also promotes senescence, a permanent cell cycle arrest, in response to oncogenetic signals, thus acting as a safeguard against neoplasia. Molecular and cellular functions of P53. P53 is a transcription factor with a modular protein dominant structure, typical of other transcriptional activators. It comprises several domains, including transcriptional activation domains, TAD1 and TAD2. These domains enhance the transcription of P53 target genes by recruiting histone-modifying enzymes, components of the basal transcriptional machinery, and coactivator complexes. Proline-rich domain, PRD, located between residues 60 and 95. This domain was initially thought to participate in protein-protein interactions, but is now considered to have a structural role. DNA binding domain, spanning residues 100 to 300. This domain is crucial for the sequence-specific binding of P53 to DNA response elements. Most cancer-associated P53 mutations occur in this domain, impairing its DNA binding ability and tumor suppressor function. Tetramerization domain, TET, responsible for forming P53 tetramers, essential for its DNA binding activity. C-terminal basic domain. This region binds DNA in a non-sequence specific manner and undergoes extensive post-translational modifications that modulate P53 stabilization and activity. P53 target genes and cellular responses. In response to stress signals, P53 activates a wide array of target genes, dictating different cellular outcomes based on the nature of stress, cell type, and environmental context. These responses include apoptosis. A P53 triggers apoptosis by inducing genes involved in both extrinsic and intrinsic death pathways, such as BAX, FAS, NOXA, and PUMA, I was about to say puma. Sensing. P53 induces sensing through genes like P21, PAL1, and PML. DNA repair and cell cycle arrest. Under conditions of lower stress, P53 promotes cell cycle arrest and DNA repair to maintain genomic integrity. Antioxidant response. P53 upregulates antioxidant genes such as SESN1, SESN2, GPX1, and TIGAR to suppress reactive oxygen species, ROS, accumulation. Metabolic regulation. P53 inhibits glycolysis and promotes oxidative phosphorylation, protecting cells from the metabolic reprogramming seen in cancer, the Wahlberg effect. Autophagy. P53 regulates autophagy, a process that can have dual roles in cell survival and death, through genes like AMPK, DRAM, and SESN2. P53 in the physiological and pathological processes. Beyond tumor suppression, P53 plays roles in various physiological and pathological processes development. P53 is involved in neural tube closure and embryo implantation, highlighting its importance in early development. Stem cell regulation. P53 restricts the self-renewal and neural and hematopoietic stem cells and influences hematopic. P53 restricts the self-renewal of neural and hematopic stem cells and influences somatic cell reprogramming to induce pluripotent stem cells, iPS cells. Aging. P53's role in aging is complex, with both pro-aging and anti-aging effects reported in different contexts. Response to therapy. P53 mediates side effects of genotoxic therapies and contributes to tissue damage in ischemia and neurodegenerative diseases. Metabolic diseases. In diabetic models, P53-induced senescence in adipose tissue leads to insulin resistance, linking P53 to metabolic regulation. The MDM2 family and regulation of P53 activity. The activity of P53 is tightly regulated by the MDM2 family of proteins. MDM2, E3 ubiquitin ligase, binds P53 and inhibits its transactivation function, promoting its degradation. MDMX, another family member, regulates P53 activity by binding to its transcriptional activation domain and modulating MDM2 function. Future directions and therapeutic potential. Research on P53 continues to uncover new functions and regulatory mechanisms, making it a promising target for therapeutic intervention. Reacting P53 in cancer models has shown potential for tumor regression, and P53 inhibitors are being explored for treating diseases where P53 activity contributes to the pathology. The P53 protein is a cornerstone of cellular stress response, with roles that extend far beyond tumor suppression. Its involvement in various physiological and pathological processes underscores the complexity of its function and the potential for therapeutic applications. Understanding P53's multifaceted roles will pave the way for innovative treatments for cancer and other diseases. Thank you for joining us on this episode of Cutting Edge. I hope you enjoyed it, and stay tuned for more explorations into the frontiers of science and medicine. Please take a look into this series that I've linked in the description. It's a book series called A Systematic Approach to Fishing. The most recent book is A Systematic Approach to Northern Pike Fishing, a data-driven guide to pike ecology, equipment engineering, and tactical fishing strategy. Again, the link is in the description, and thank you all for watching this video.